Okay, so in the case of the regulation or transcription, what we are going to learn is something which you already know. That is like already we have talked about, or maybe you have studied about the lac operon, right? So how this operon is getting, regulated, why this is important in the case of the prokaryotes, that is what we are going to learn in the regulation part. Okay, so already you know that there are certain Z, X, Y genes are there, but which gene is getting expressed first and why, what is this leaky expression every time? Okay, now why this leaky expression is important, why it is also of disadvantage. So this is what is important with respect to the exam. Okay, so this is something which we are going to learn. So before that, there are many certain things which we need to understand before we proceed. Okay, so let's start with transcription. So what is the principle behind transcriptional regulation? So for all transcription regulation, we have got, there are certain proteins called the regulatory proteins. Okay, now these regulatory proteins are nothing but two types are there. Either you can have the activating proteins, okay, or you can have the repressor, which is going to inhibit the activity. Okay, in short, they are the activators, they are the repressors. Okay, now these activators and repressors will get in action only depending on the extracellular communicating signals. Okay, this is not within the cell communication, this is from the outside, then only these activators and repressor will get they will come into action. So typically these regulators are DNA binding proteins which recognize specific sites or near the genes they control. Okay, now activator is going to increase the transcription rate, whereas repressor is going to decrease the transcription rate. Now this is really interesting once we come to lac operon and how this is happening, because there is this cis and trans ways by which this activator and repressors are actually inhibiting, okay, or they are activating. Now most activators, once again, most activators and repressors, they act at the level of transcription initiation. Now just think about it, like why it is initiation, क्यों नहीं बाद में elongation या termination में करते हैं, right? This is something, like किसी भी बंदे को आपने कुछ काम दिया है, वो टर्मिनेट करने वाला है, जैसे ही उसने काम खत्म करने वाला है, एंड तब आप उसको बोलोगे दैट इस काम की जरूरत नहीं है, जस्ट इमेजिन हाउ दैट पर्सन इज गोना फील, राइट? दैट मच टाइम, दैट मच एनर्जी, एफर्ट, एवरीथिंग इज वेस्टेड. So this is what a cell understands कि ये मुझे नहीं करना है, and that's why these regulations are there. So if a cell understands that this particular mRNA or protein synthesis is going to be wasted because it has got mutation or it is truncated, so I'm not gonna go further, मैं उसको पहले ही stop कर दूंगा, ठीक है, so that I don't waste my energy, I don't waste my time. Okay, so that's why many these activators and repressors, they are going to act at the level of initiation itself. Now initiation, so the first transcription initiation is the most energetically efficient step to regulate. By this, what we mean is that deciding whether or not to express a gene at the first step ensures that no energy or resources are wasted. Now, what kind of resources are they talking about? Energy, so we understand in the form of ATP and GTP, right? Now, what resources do they mean here? Akshaya? Yes?
The proteins.
We are talking about transcription. So the NTPs, okay? Eventually, if you're going to degrade that mRNA or rRNA, why to waste NTPs? Correct? Because see, NTP क्या है? Triphosphate है. So when it gets inserted or incorporated into the mRNA or RNA, what happens? 2 PI is released, that energy is getting wasted, right? Again फिर हमें इसको energy देना पड़ेगा so that ये NTP बने दोबारा से. So all these resources are getting wasted, right? Not only the NTPs, the other cofactors also, magnesium हो गया, RNA polymerase हो गया, everything which is involved in this. Now, second, the regulation at this first step is easier to do also. So there is only a single copy of each gene. अब just imagine अगर एक mRNA है उसको रेगुलेट करना ज्यादा इजी होगा एज कंपेयर टू एक mRNA से 1000 प्रोटीन्स आ गए, राइट? इफ सपोज लेटर द सेल अंडरस्टैंड्स कि अरे ये गलत mRNA था, इससे जो भी प्रोटीन आए हैं दैट नीड्स टू बी रिप्लेस इन द सेल. So which is better? Either to degrade the mRNA or degrade the 1000 proteins. तो कौन सा easy and कौन सा cheap होगा? Of course, the first step, right? Where you can degrade the mRNA to stop the further expression. Okay, so this is why the transcription initiation is the best step at which the regulation should happen. Am I clear on this part?
Yes, ma'am.
Okay. Now, same thing. So here, RNA polymerase is a, so you know that it binds to the operator, sorry, it binds to the promoter, and operator lies in the regions where promoter is always present. Now, what is this operator? Operator is a region where your repressor will come and bind. Okay? Activator has got the site differently. Now, later, we are going to see that there is RNA polymerase alpha CTD. If you remember, this is the alpha C terminal domain of your RNA polymerase, which goes and binds to the activating region, CAP region, catabolite activated promoter region. We'll come to that, don't worry. So this is where the activator will bind, and this is where the repressor will bind. So you know that if everything is good, the basal level of transcription will happen. Now, if repressor goes and binds to the operator, what will happen? The RNA polymerase cannot bind to the promoter. Hence, no transcription. Now, RNA polymerase is there, which goes and binds to the promoter, plus with the help of the activator, the level of transcription or the rate of transcription is enhanced. Clear? Simple logic. Now, let's make it more complicated soon. Now, this is something which you need to understand, that these activators, these repressor, either they can present like this, okay, side to side, or if the regions are far, they can bind over there. They can bind to the different regions. They can make the loop and come together for the interactions. Okay? Now we'll see. 1 second, guys. Huh? Okay. So as I was saying here, so whenever these activators or repressors, if they're present far also, if these sequences are present far also, then also they come bind over here and come together. Okay, so these are the mechanisms. So keeping this thing in the mind, in later in the later lecture, we are going to see how activators and repressors are actually regulating all this. Okay. Now, the cooperative binding and the allostery have many roles in gene regulation. So look at over here. See, here you see that if suppose your activator sequences are not present here, okay, if suppose it is present somewhere far, but then how it is going to help in increasing the rate of transcription? So this is how. So the activators, the DNA will loop out, it will interact with the RNA polymerase so that the transcription take place. But then looping of the DNA, it's not random, okay, it needs another DNA bending protein, and that's how it forms a bend or loop. So DNA loop out. So what is happening? This activator will come and bind to the RNA polymerase. Clear? Okay. Now, let's talk about the lag operon. So we know that this is about the lactose metabolism in E. coli. Now, one thing what you have to understand here is that E. coli, the main food source is glucose. Okay, even for humans also it is glucose. So whatever we eat, it gets metabolized to glucose, and this glucose goes inside into our cells, right? In the same way for E. coli also, it is the glucose. Now, if suppose the glucose is not present, then lactose is there, right? This lactose is what? It's a disaccharide. Okay. Now, who can tell me lactose is made up of? Yes. Anyone? Tanvi? Lactose is made up of what two sugar units? Akshaya?
Glucose and galactose.
Correct. Now, here also, so lactose is glucose and galactose, right? Now, so obviously when lactose gets metabolized, glucose will be consumed. Correct? What about the galactose then? So do you guys know what is epimers? Yes? Do you know what are epimers? Anyone? No? No? Okay. So epimers are nothing but they are the glucose having they have got the same glucose structure. Okay. It's just that at that particular position. For example, if I say mannose, okay. Mannose is epimer of glucose. It means that at the second carbon, instead of HOH, they have got OHH. Okay, this is the structure of glucose if I make. So at the second carbon, it is the HOH, OHH ka ulta ho jayega. Same way, galactose is 4 carbon epimer of glucose. Okay? So see, you will be able to understand more if you go through the structures, and it is going to help only to understand. So just go back, brush up your little basics of biochemistry, like how you write the open structure of glucose, what mannose is, what galactose is. Okay, so it becomes an easy. So then we have got epimerase enzyme. Okay, they are going to convert galactose into glucose so that it will get consumed. Otherwise क्या होगा? E. coli cells में galactose इकट्ठा होता रहेगा, right? And eventually everything which is in excess becomes the toxic. So it's good that epimerase enzymes are there and they convert galactose into glucose. Clear? Now coming back to the lac operon. So it has got the 3 genes Z, Y, A, and they're arranged adjacently on the E. coli genome. So they are like operon. Why? Because they have got all the gene together and they are known as polycistrons. So I hope everyone knows this thing. Now look it over here. This is your promoter region. This promoter region is overlapping with the cap site. This is your activator site, okay? And this is a promoter operator site where the repressor is gonna bind. Is this much clear so far? Good. Shruti, clear?
Yes, ma'am.
Yeah. Okay. Now about the genes. So LAC-Z, you know, it is beta galactosidase. Okay. Now this is going to cleave your lactose into galactose and glucose. And eventually you're going to use glucose for your energy source. Later galactose is converted to glucose with the help of the epimerase enzyme. Now, Ysine is lactose permease. Do you guys remember lactose permease हमने कहाँ पढ़ा था? In cell bio transporters. So there we have studied about the lactose permease. It is actually a transporter which will take, so it is present on your E. coli membrane. So it is a transporter actually which will take your lactose from outside to inside. Okay? Then this beta galactosidase will convert your lactose into galactose and glucose. Now then we have got LAC8 gene which encodes thiogalactoside transacetylase. Okay, so this will get rid the cell of toxic thiogalactosides. As such इसका role operon में क्या है नहीं मालूम, but we say that to avoid the toxicity this enzyme is present. Okay, so now these genes are expressed at high levels only when lactose is present and glucose is not present. Okay, अब यहाँ पे सवाल आता है, since the operon will only get activated in the presence of lactose, right? But lactose तो cell के अंदर लाने के लिए आपको lactose permease चाहिए, which is a Y gene product. But this will get only activated when lactose is there. तो ये जीन पहले से प्रेजेंट होना चाहिए ताकि वो लैक्टोज को अंदर ला सके. Now, how is this possible? Is my question clear to you?
Yes, ma'am.
So what do you think? What is the answer?
Maybe the presence of lactose causes a chain reaction that causes lactose permease to come in, so that lactose can get in.
No, Shruti. See, once the lactose come inside, this galactose glucose is formed. Okay, then only see once lactose comes inside, there is a conversion of lactose into allolactose. Now this goes and binds to your lac operon, and then only these genes gets activated. Okay, but to bring lactose inside the cell to get converted into allolactose, you already need the presence of lactose permease. पर ये कब आएगा जब lactose अंदर होगा, but lactose अंदर लाने के लिए आपको lactose permease चाहिए. So you see the irony over here, right? So here the expression, so here what is something known as leaky expression. Okay, so this concept is there. So leaky expression says that according to this, the lac operon is not tightly regulated. थोड़ा सा express होता ही है. So you have got few proteins already expressed. ठीक है? In the E. coli. So that's why these lactose permease in few copy numbers are present. And when lactose is there, it comes inside. Then your lac operon gets activated. And more copies of lactose permease comes in. So that whatever the available lactose is there gets utilized by the E. coli. Now it is clear?
Ma'am, is there a difference between leaky expression and basal transcription of these genes?
Yes. So basal expression is when there is a signal given to the cell that you synthesize this particular gene. Okay. Now, depending on the presence of the activators and repressors, either increase or decrease rate of reaction is there. That is basal level. जहाँ पे आपको cell ने बोला है, signal दिया है कि synthesize thousands of proteins or thousands of mRNAs. ठीक है? Leaky expression is that it is blocked. There is no signal. But still, RNA polymerase has synthesized certain mRNAs. There is a certain translation happening without the cell knowing. समझ रहे हो? This is
so during basal expression, lactose is present and it's the cell is producing all of these genes at maximal rate during basal secretion.
Yes. Leaky is not tightly controlled. ठीक है? Leaky word says what? Even though you have tied the tap, still the water is dripping. So that is a leaky flow of water, right? In the same way, here also the expression is leaky. यानी कि क्या एक दो mRNA बन गए हैं, एक दो protein synthesize हो गए हैं. And this is also required so that in future, if the E. coli is not having glucose or lactose दे दिया है, तो उसको lactose पे survive करना होगा. Right? See, this is same thing. If you don't like eating karela, but अगर हम आपको बार बार वही दे रही हूँ, so what will happen? Over a period of 2-3 months, you will start loving eating karela. So in future, you won't be scared of it, right? It is like that. Okay, back to here. So now there are two regulatory proteins called CAP and the other one is the lag repressor. Okay, so lag repressor is by the lag I gene, which is located near the lag genes but transcribed from its own promoter. यानी कि क्या lag I genes का खुद का promoter है. Now cap is for your catabolite activator protein. It is also known as CRP for CMP receptor protein. क्यों? Because later we'll see that the involvement of CAP, that is CMP, goes in binds to the catabolite activator protein in order to enhance the activity of transcription. Okay? Now the gene encoding cap is located elsewhere on the bacterial chromosome, which is not linked to the lag genes, but they all together. इसलिए हमने बोला था DNA loop formation is important because that's how the RNA polymerase will get activated. ठीक है? Now both cap and the lag repressor, they are the DNA binding proteins. So they are going each. So each will bind to a specific site on DNA or near the lag promoter. Okay. Now let's see if I have the figure. Okay. See here in this figure. So you have got the RNA polymerase. You have got the RNA polymerase, right? Now, if everything is there, glucose is there, lactose is there. So what is happening? The RNA polymerase will go and bind, basal level expression will happen. Now, repressor, you don't have lactose, you have glucose. So no transcription. Now, glucose is not there, lactose is there. So what is happening? Cap, the activator is there. It is helping your RNA polymerase. So activated level of transcription is happening. Okay, clear? The same thing is written over here also. You can just go through later. Is this figure clear? ये थोड़ा और complicated होगा आगे जाके. Shruti, any doubt over here?
Yeah, the basal and activated levels.
Acha. Okay.
So the basal.
Okay.
Yeah, go ahead.
I'll explain. Okay. I think I got your confusion. Okay. See, if let us say if only glucose is there. So, Shruti, you do understand that if glucose is there, I don't need lacoperon to get activated, right?
Yes.
Okay. This is that scenario. And that's why the repressor is there, which goes and binds to the operator. There is no transcription. Okay. So, this is everything with respect to lacoperon I'm telling. Now, glucose is not there, lactose is there. So, you do understand activated level of transcription, correct?
Yes. Yeah.
Okay. Now, the last scenario where you have got lactose and glucose together. Okay. When both are present. Now, what happens is that, Shruti, glucose is there, lactose is there. So, the first preference of any organism is to utilize glucose.
Glucose.
Correct? But because of leaky expression, okay, the E. coli had 1 or 2 copies of your lactose permease. Okay. Because of leaky expression, lactose permease is present. Because of this presence of lactose permease, the lactose was able to get inside. It was getting converted to allolactose. This resulted in low level expression of your lacoperon.
Okay.
ठीक है, अगर 100 ग्लूकोज थे और 100 लैक्टोज थे, सो ई कोलाई ने ये पूरा यूटिलाइज किया और यहाँ पे कुछ 20 यूज किया बिकॉज ऑफ दिस बेसल लेवल एक्सप्रेशन.
Okay.
Okay. Now, in other words, there was no glucose and 100 lactose was there. So starting में उसने 10 use किया because of the leaky expression. Now once lacoperon got activated, all rest 90 were also used. अभी clear है?
Okay. Yes, got it.
Okay. What about others? Akshay and Tanvi, clear now? The basal level, no transcription activated, right?
Yes, ma'am.
Okay.
Yes, ma'am.
Cool. Now this is the entire thing. ये समझ में आ गया तो सब समझ में आ गया. Okay, so again यहाँ पे इंड्यूसर क्या हुआ आपका? इंड्यूसर क्या है? एलोलैक्टोज. लैक्टोज, एलोलैक्टोज. ठीक है. Now in the absence of inducer, okay, there is no lactose, there is no allolactose. What is happening? mRNA repressor has been synthesized. So repressor goes and binds with the operator. There is no lac operon activation, यानी कि ट्रांसक्रिप्शन नहीं हो रहा है. ठीक है, क्योंकि वहां पे नहीं है आपका lactose या allolactose. Now in the presence of the inducer, what is happening? In the presence of the inducer, जो आपका repressor है, okay, now repressor को आपका allolactose bind करेगा. So what will happen? There will be a conformational change. Because of that conformational change, the repressor is no longer able to bind to the operator. So operator is now free. Hence, RNA polymerase will go and bind over here, synthesizing these mRNAs, resulting in the expression of galactosidase, permease, and trans-acetylase. Clear? Simple, right?
Yes, ma'am.
Yeah, I'll just repeat once again. So, लैक्टोज अंदर आने के बाद एलोलैक्टोज में कन्वर्ट होता है. ओके, नाउ दिस गोज इन बाइंड्स टू योर रिप्रेसर. So the repressor is no longer bound to the operator. It is free. So RNA polymerase यहाँ पे आएगा. It will synthesize or it will continue the transcription. Okay, cool. So everyone okay with the lac operon? Confidence is there. Now in the next lecture, okay, in the next lecture there's something about, I want you guys to understand or read and come like what is haploid. I'm sure you know what is haploid. Diploid. So same way triploid, tetraploid and meroploid. Okay, so when I talk about lac operon in the case of haploid, diploid, meroploid, how the lac operon is going to behave differently in all these cases, we're going to discuss that. Okay, so before that, please study and come. What are these or what is the difference among these? Okay, I just made this title only. Okay,